Chronic obstructive pulmonary disease, or COPD, is the third leading cause of death worldwide. It is an obstructive lung disease characterized by long-term poor airflow caused by chronic bronchitis and emphysema. Chronic bronchitis is the continued inflammation of the bronchial tube lining, and emphysema is the destruction or damage to the alveoli, or the air sacs in the lungs, where they get stretched out and are unable to properly function. The respiratory alveoli are constantly exposed to external irritants, which are naturally mitigated by cell stress responses. Mir-24 is a small non-coding RNA molecule that regulates gene expression. It has been shown to regulate the expression of two vital cell cycle control genes. This research will be done to see in what way Mir-24 affects cell behavior and viability of cells exposed to cigarette smoke extract, modeling the lung's response to a specific oxidative stressor. It was hypothesized that when exposed to cigarette smoke, or CS, cells with overexpressed Mir-24 will respond with high percentages of dead cells. This will be tested by culturing 4 sets of BS2B cells, which are lung epithelial cells. There will be a control with no added CS, a control with 1, 2.5, and 5% added CS, a mimic control with overexpressed Mir-24 and no added CS, and a mimic test sample with 1, 2.5, and 5% added CS. Thus, the acquired data can be compared along all potential variables. Cigarette smoke extract augmentation and quality will be consistent through all applicable samples. It will be acquired by using a cigarette smoke extract apparatus and running the smoke through a media that will later be added to the cells in different concentrations. All petri dishes will be supplied with an equal amount of standard media, RPMI 1640, to provide the necessary nutrients for growth. A cell count will be done every other day to identify the cell growth curves and percentages of dead cells. To do this, a hemocytometer will be utilized, as well as a trypan blue assay. It will dye blue the dead cells in order to yield comparable percentages. The culturing conditions, temperature, nutrition, and time will be kept constant for all the samples. The number of cells dyed blue by the trypan assay will be indicative of how many cells died at various concentrations. Preliminary results support the hypothesis. This research will contribute to the goal of seeing what precise mechanism contributes to the development and exacerbation of chronic obstructive pulmonary disease. Being such a frequent cause for death, understanding of the mechanisms is crucial for counteracting its effects. This research will be done with the approval of Dr. Mir-24 and the guidance of Eric Finmore. It will take place at the Yale School of Medicine.